Welcome to Omega Matters, where we talk all things omegas. I'm Kristina Jackson. This is Bill Harris. And today we have a great guest, Dr. Kevin Klatt. Kevin is an assistant research scientist and instructor in the Department of Nutrition Sciences and Toxicology at UC Berkeley. He received his PhD in molecular nutrition from Cornell University and completed his dietetic internship at the NIH Clinical Center. He is also the inaugural Dennis M. Beer Young Career Editor for the American Journal of Clinical Nutrition and hosts the AJCN podcast called AJCN In Press. Kevin has experience from the lab bench to human clinical trials to clinical practice, and we are very excited to talk with him about his work in the omega-3 space. So thank you for joining us today, Kevin.
Thank you so much for having me.
Yeah.
I do have to note, unfortunately, I'm no longer young, so they kicked me out of the young career editor position, and I'm just an associate editor.
Well, congratulations still. AJCN's kind of the big dog in nutrition, so
at least they didn't kick me out completely.
Yeah. you got a promotion. Can you
young,
yeah. Can you take us through a little bit of your career journey? How did you get into nutrition in the first place, and how did you get to where you are now? I know it's a lot, but yeah.
Yeah, I mean, that's taking you back. I actually wasn't really into nutrition until, I guess early in college, I got into it. I had lost some weight in high school, and, that exposed you to the whole world of what's the best diet, yada, yada, yada. But I went to an undergrad that didn't have a nutrition program, and I had a scholarship there, so I didn't want to leave.
Mm-hmm.
So I did a relatively long and winding path as a biological anthropology major, which I think is a great undergraduate major. And it was, I think understanding human variation and human adaptability is super important context in nutrition, especially where things get dogmatic and prescriptive really fast. But I did all the classes I could that would sort of, get both the dietetic and the sort of nutrition science background. I did a quick year at Drexel University. I'm from Philadelphia, went to undergrad there. So I did some of the dietetics coursework there. And I was just really eager at that point to get into research. My honors undergrad thesis advisor or just advisor always told me, she's like, you're going to be a researcher. And I was like, no, I want to be a clinician and practice. She's like, all you do is ask questions about how we think we know this and how do we at knowing it. And I was so vehement that I wasn't going to be a researcher. And I sent her an email a few years ago being like, oh, you were very right.
Yeah.
Listen to your undergraduate advisors. They know you better than you know yourself.
Oh, yeah.
But I was eager to get into research, but I also wanted to finish the RD. And so I just applied to a couple programs where I had 2 DPD classes left that I needed to finish. And I was too eager to get into the full research track and things. So I did my PhD at Cornell in Marie Caudle's lab, and she's who I went there to work with. And, this is so 2013 coded of me, but I was really interested in epigenetics at the time.
Yes.
And particularly just the idea that the methyl donors in the diet and the amount of them could influence histone post-translational modifications in CpG, methylation state, and influence gene expression was just this wow. And I look back on it being oh my gosh, we have so little molecular tools, both at the cellular and in vivo level, to really say anything too causal. And there's a bit of naivety I look back on, but I'm still fascinated by that idea of what you eat during pregnancy is structurally building a fetus. It is laying out what their metabolic state is going to be in some ways for the rest of their life. And so Marie had done a little bit of work in that epigenetic space, but choline was also interesting to me because, particularly in pregnancy, because the power, potential, and promise of epigenetic programming, or whatever, and then you look at the DRIs and they're oh, a study done in men was used to estimate the requirements in non-pregnant women, and then they adjusted the non-pregnant women to study to get the pregnancy value, and you're okay, you don't have to be a feminist to say maybe pregnancy DRIs don't have to come from studies done in men. So there's a lot of work that needed to be done, clearly. And Marie was doing a lot of that work. She and Steve Zeisel were the 2 big folks in the field. So, and Marie had the kind of PhD RD training. And so I very much looked up to her from afar and was fortunate to get to work in her lab. And so I ended up doing when I was with Marie, one of the my big career goals was to, I think what I found most frustrating, people are like, what'd you go into your PhD to do? And I just wanted to build like a toolkit. Like I didn't really, as much as choline seemed like an opportunistic nutrient, but I didn't want to be a single nutrient person. Like at the time, the vitamin D wars were going on, and people are screaming at each other at conferences about vitamin D recommendations. And I was like, I just don't care about any nutrients to go scream about at the conference.
Yeah.
And I was just very interested in I was reading papers. I was frustrated if I read a cell culture paper or I read an animal paper. I could get it, but I didn't know how to critique it. And I think you really have to do the work to understand all the nuances of critiquing it. And so I set out really just to try and get as many skill sets under my belt as I could. And Marie's lab was really great for that because she very much encouraged her students to go work in another lab for a year and do some of the students that were doing whole body human metabolism with vitamin D also went and did some work in a cell culture lab where they were studying vitamin D in a placental cell line there. And so that was really great. And so I ended up having a co-mentor, a reproductive biology co-mentor, Mark Roberson, and spent a bunch of time in his lab. So I got the full gamut of, he was doing cell culture and primary cells and knockout mice. And then Marie was the sort of whole body human physiology, isotope tracing, LCMS lab. And it was a lot of hours of work to be across two labs, but it got me that goal of really wanting to work across the translational spectrum. And then after I did my PhD in Marie's group, I was doing a big RCT that we'll talk about the results of today. And then in Mark's group, I had my hands in a number of projects, but I had a sort of spin-off in nutrients influencing some of the nuclear receptors, which are transcription factors that regulate gene expression, and hunting down a potential ligand. And that's some of the work I continue to do today, in addition to the human clinical trial stuff in nutrition. And so after I left Cornell, I went and wrapped up the dietetic internship, and which was nice. I highly recommend to take a break from research, especially after your PhD. You're a little burnout. Did my clinical training, and then did a postdoc down at Baylor, kind of following up on the more nuclear receptor stuff. But I had the clinical trial data coming in from working with Marie and the fatty acid analyses that we did together, and we'll dive into. So I've tried to stratify this, both a little bit of human trials and a little bit of basic science for most of my career, kind of, I guess my entire academic career at this point, which is increasingly hard to do. But yeah, that's kind of me in a nutshell. I've covered different nutrients from choline, did a little bit of B12 work, and continue to do that. And then different fatty acids, both some of the saturate, there are shorter saturates like laurate that are kind of fun and interesting, and then some of the omega-3 work as well.
You have always been someone who I think just kind of blows me away with, more about more things than I can even speak about. And I've been out of grad school for long enough and in one lane where I'm starting to feel like, oh, I can't talk about anything else besides fatty acids because I haven't been keeping up at all. All right. So, but it's always fun to hear you at conferences just come up and just bring that little bit of a more holistic type of view when we get too narrow and focused in on things. And that's where I do think where your work with the choline and the DHA in this RCT you did at Cornell, it's one of the first that we've seen where omega-3 levels get kind of affected by another nutrient in a very specific time frame, which is the caveat. But so I would love to dive into this part of the talk. So let's go into the science. Let's go into these two studies that were run. I don't know how much you want to talk about both, but the paper for everyone that wants to check it out is the one that you're lead author on is Prenatal Choline Supplementation Improves Biomarkers of Maternal Docosahexaenoic Acid Status Among Pregnant Participants Consuming Supplemental DHA, a randomized controlled trial. That is AJCN in 2022. Is that the primary one? Sometimes there's several. Okay. So can you kind of set the scene and tell us what you
For
did.
sure. Yeah. So I was always eager to not be a single nutrient person. So this was sort of like the perfect topic. And Marie was very interested in the choline omega-3 fatty acid sort of interactions. And the rationale for it, which it gets a little metabolic biochemistry, right up Bill's angle for sure. Um, but so like supplemental DHA in the diet, it can come in various forms. Like we don't really eat the free fatty acid form. You get it typically triglycerides. Or you can eat these phosphatidylcholine forms. But for the most part, it gets deconstructed in the intestine, absorbed, and the body is repackaging it to the way that it needs it. And you can find it in circulation, esterified in different forms within lipoproteins or circulating as a free fatty acid bound to albumin or as a lysophosphatidylcholine bound to albumin as the primary ways it's getting around. And so there's lots of debates in the literature about what circulating form, particularly like in pregnancy, is there for the fetus, for the adult human brain. There's all these big debates right now about whether the free fatty acid form is sufficient or how much these LPC DHAs sort of supply the adult human brain. But within pregnancy, one of the things that's really interesting that starts to link choline and omega-3s is phospholipid synthesis is dramatically increased. And so normally we think about DHA getting incorporated into phospholipids, particularly phosphatidylcholine. And in pregnancy, there is the gene that produces phosphatidylcholine de novo that gets dramatically upregulated in pregnancy is called PEMT, or the phosphatidylethynolamine and methyltransferase pathway.
Whew.
But it's taking, so we'll call that PEMT,
Yes.
but it takes phosphatidylethynolamine, another phospholipid, puts 3 methyl groups on it to make phosphatidylcholine. And early work back into the 90s, and then the knockout mouse for the PEMT gene in the 2000s really identified this link that the PEMT pathway produces a phosphatidylcholine that's particularly enriched in omega-3s like DHA. And so that starts to form the linkage between choline metabolism and omega-3 fatty acid metabolism. And so when you knock out PEMT in mice, when you do like fatty acid profiling of their plasma, the DHA is like dramatically reduced in the knockout mice relative to the wild type. So we think, and PEMT, I should say, is very highly expressed in the liver and not really anywhere else. And so we think that this is a liver-specific pathway for getting out, for synthesizing phosphatidylcholine in general, this major way that the liver exports omega-3s. And in the pregnancy...
Can
Oh, yeah,
I
go for it.
see if I'm tracking this? So we consume omega-3, the body breaks down, absorbs the fatty acids, the liver remakes it. And most of the fatty acids that would go to the placenta are this specific kind of phospholipid, phosphatidylcholine, that is attached to albumin, or is it within other...
Phosphatidylcholine will be more in the lipoproteins.
Okay.
The LPC is bound to the albumin, yeah.
Okay. And so people are tracking that specific, because there are lots of, so when we talk about omega-3s, we're talking about red cells, but red cells are also, those are phospholipids as well, just all different kinds. And so this is a specific kind of phospholipid that seems to be the one that is getting across the placental. That's the one that goes in the placenta, or does it kind of...
We think it's really important for getting from the liver into circulation. And then from circulation, it's going to go to all other tissues, including the fetal compartment.
Okay.
So in just the general adult, though, this pathway of producing phosphatidylcholines enriched in DHA, this gene, protein PEMT, is really important for getting that out. And the pregnancy link comes in that there's, and molecular biology speak, there's a promoter in front of the gene where estrogen is able to bind and upregulate the expression of the gene. And we know across pregnancy, there's like a 10-fold increase in estrogen. And so Marie's previous study had shown that with stable isotope tracing, that PEMT activity was like really increased across pregnancy. And that sort of fits. You want to be able to produce a lot of phosphatidylcholine to build this baby with all its cell membranes. And what's interesting is that, during the 3rd trimester of pregnancy, you have lots of brain development and eye development where you're depositing these PUFAs. And that's also the time at which estrogen peaks the highest. So you can sort of form this like evolved axis where estrogen's increasing so that the maternal liver produces a ton of phosphatidylcholine and a ton of PUFA, gets them out to support fetal brain development and just development in general. But one of the things that we questioned was PEMT, it's a methyltransferase. It uses tons of methyl groups, and it's known to be one of the largest consumers. You have hundreds of transferases in the body, but quantitatively, as far as it goes for methyl group consumption, it is one of the major ones, and it's probably the major one in the pregnant body. And so it needs a lot of methyl groups, and we wondered whether that reaction was potentially limited by the fact that, coincidentally, choline is the major methyl donor within the diet, and choline itself becomes so important. It is really concentrated within the fetal compartment. And what Marie had seen in her previous study is that of all the metabolic fates of choline, it seemed like when you had lower choline intakes, there was less oxidation to betaine. And that is the committed step for using choline as a methyl donor. So there was a shunting away from its use as a methyl donor so that you could concentrate choline within the fetal compartment and support other phospholipid synthesis pathways. So at this time, when you have the most consumption of methyl groups, you have a less utilization of choline as a methyl donor. And so we wondered whether giving choline back would supply those methyl groups to basically fuel the maximal activity of PEMT. And so whether it was essentially rate limited by not having enough dietary methyl donors, because metabolically you're so stressed with choline during pregnancy, it's being reprioritized away from that methyl sort of metabolic fate and utilization. So because at a high level, we're thinking that the PEMT activity is important for just getting out phosphatidylcholine enriched in DHA into circulation in general in all humans. But in pregnancy, that pathway is dramatically upregulated at a time when you're also using choline less for generating methyl groups that's required for the PEMT activity. And so we just asked simply if you supplement with choline back, which you think has independent benefits on its own, particularly for fetal development, that the choline supplementation would be beneficial. And Marie had seen that in a previous study, that you might also have these metabolic benefits and enhance the utilization of DHA. So we designed a study where everybody got their prenatal on both arms. Everybody got their standard of care at 200 milligrams of DHA, but then one group got a small amount of choline in just the form of a stable isotope tracers with 25 milligrams per day. And then we also gave 500, the other group got 550 milligrams of choline, 500 from a supplement, and then 50 milligrams of the stable isotope tracer. So this mirrored very much what Marie had previously seen in her controlled feeding study she did before, that high doses of choline just independently improve fetal cognition. And so we're still doing, infant cognition. So we're still doing the postnatal follow-up with Rick Canfield and Barb Strepp do that. We do all the pregnancy and metabolic element of that, and then we sort of pass off the infants to our human development colleagues who then assess these sort of really important, I think, somewhat clinical outcomes of cognitive development. So we try and have these are tons of work to do these studies. So we try and get all the outcomes that we can that are relevant to choline. But our primary outcome for this was really looking at this choline DHA metabolic interaction.
Yeah.
Is there any
Can I ask an RD
data
question? Go ahead.
Does choline do anything to enhance the synthesis of DHA from ALA?
We have there's not really any data on that I'm aware of. It's possible. The metabolic theory, I think, is kind of lacking on it, but nobody's ever looked in giving an ALA sort of tracer alongside different doses of choline and seeing if you enhance DHA synthesis. But I don't know of it. I'm trying to think of any data that would exist to even imply that. Most of it's been this context of Steve Zeisel has done some work where in choline deficiency in premenopausal women, they also see reductions in circulating phosphatidylcholine DHA just outside of the pregnancy stage. So most have looked at if you compromise omega-3 choline and methyl donor availability in individuals that have really high PEMT activity, then you might limit DHA availability that's out there.
Right. Okay. That's kind of because we have seen estrogen does like affect the pathway to increase DHA, like they've given. So, but is that at all like connected to the choline story? If that's also how it's like how we end up measuring it, I don't really know if there's a separation of the two, if that makes
I
any
think it's
sense.
not right now. It's like estrogen influences ALA to DHA, and then estrogen
Yeah.
influences de novo choline production through the PEMT pathway. Whether they synergize in some way
Yeah.
that's not fully known, but we think they're relatively independent effects.
Hmm.
So the estrogens, that's basically prioritizing the
Hmm.
de novo synthesis of both of these really important nutrients.
Which is
Um,
so
How
cool.
much?
Like
How
that
much PC?
pregnancy is taking care of that in some ways.
Yeah.
It's cool.
How much? How much PC do we eat? Do
Yeah,
we eat
that was my
much?
question.
Oh,
Or
okay.
just
You didn't ask
choline.
it then.
This is a
How
RD
much?
question.
Well, it's an RD
yeah,
question. Well, phosphatidylcholine,
Choline
phospholipid.
or
Do
choline.
we need to make more than we eat? Apparently, even of PC.
Yeah, we make a ton of PC. So it's for a while, people didn't even think choline was a essential nutrient because this pathway exists. So you can use other dietary methyl donors like methionine or betaine to fuel the PEMT activity, and you take phosphatidylethanolamine and make phosphatidylcholine, and you have phospholipases that can cleave that choline head group off. So there's it's somewhat akin to vitamin D as an example, not obviously the same, but you can make some, and it took proving that we're totally removing choline from the diet while keeping the intakes of folate, B12, methionine, all like still high that facilitate the rest of those methyltransferase activities to show that there's still some clinical consequence of this. So you need to eat some amount of it. The exact amount is still not known. So there were seminal studies that Steve Zeisel did, basically giving everybody a run-in diet with around it's like 7 mgs of choline per kilogram. So it ends up being about 550 milligrams that folks got, and then really depleting to like less than 50 milligrams and watching for whether folks get signs of fatty liver. And it also appears that there's a muscle damage phenotype that can occur in some individuals, and
Mm.
then repleting them back. And so in later studies beyond when we had the DRIs in America set, there was more of a stepwise reintroduction of dosages to kind of identify at what point do we meet the needs of like 50% of the population.
Mm.
But the DRI is really based on that 550 value that they did in the depletion repletion studies. Um, and so, yeah, you basically, it's recommended, the AI that is given for the DRI value is 550 milligrams for men, and then a body weight based adjustment for women.
Mhm.
It's a little bit less, and then a small additive increment of 25 milligrams for pregnancy back onto the women of reproductive age
Mhm.
values. And so
Mhm.
it is a big guessing game. And in DRI speak, for folks who don't know, the AI, it means adequate intake. But I always teach this when I teach it to students. I would say when you hear adequate intake, you need to think inadequate data,
Yeah.
because it means there wasn't dose response data available to estimate the average requirement of a person, and then go 2 standard deviations out to set an RDA. So there's a lot of uncertainty here, but you eat essentially a few hundred milligrams of choline on average a day, and egg intake is a really rich source of phosphatidylcholine. So most of the choline you're eating, like 75% of it in the diet, comes in as PC. About 25% comes in through other forms. So when we say dietary choline, it's kind of like it's not a single compound; it's actually
Okay.
several choline-containing compounds.
And it's
And then
not
when in
free
our
choline.
trials, we use.
It's not
What's that
free
again?
choline. It's not free choline. It's always esterified to something.
Very low.
Yeah.
No, there's a little bit of free choline that you'll absorb in the diet, but it's pretty minimal. We use choline salts, which will come in predominantly as a free choline
No.
in our supplementation. So that's a big kind of caveat where it's really hard to back translate from supplements to what it means for food with choline, because we're not giving the form
Yeah.
that is mostly found in food, and that's largely because phosphatidylcholine is only like a 12% choline by weight. And so when you do an intervention study, you've got to ask, and this has happened in pregnancy studies, where you've got to ask women to take like an extra 7 or 8 kind of horse pill size things on top of their prenatal, on top of the DHA. And
Ooh.
so you always worry about compliance and burden, um, just on the participants. And it also scientifically becomes an issue of which form of phosphatidylcholine do you give, because you start to give meaningful amounts of the fatty acids that are on the phosphatidylcholine molecule. And so you're co-intervening not only with choline, but like the linoleic acid that comes in with the soybean oil, phosphatidylcholine. And so it's a design nightmare. And so
Yeah.
giving the choline salts is much easier to work around. They do taste gross. I have sat at a metabolic kitchen test tasting when you put 500 milligrams of choline. Like it turns out black tea actually really masks a lot of the bitter and sour taste of the choline salt. We gave it in a small juice cocktail. Like we made 15 mil Eppendorf tubes, and people just took a shot of choline. And sure, we could, assess compliance and things that way and make it easy for folks. But yeah, it is. There's so much like dietetic angles on this. I could
Oh
go
my
down a whole rabbit
gosh.
hole, like
Yeah.
how you translate this into population level recommendations. But for and I should say the metabolic fate is probably reasonably different between a choline salt, which is water soluble, comes into the portal vein, hits the liver. A lot of it looks like it gets oxidized to betaine because that's what the liver does when it gets tons of choline at once. Whereas phosphatidylcholine is going to come in and largely gets repackaged into chylomicrons and goes through that route where a lot of extra hepatic tissues are going to see it first before it makes its way back to the liver. And so there's a little bit of isotope tracing data saying that the initial metabolic fates are different. Whether that has long-term consequences, we don't really have great data in humans comparing head-to-head different forms of choline. But there's enough, I think, biochemical rationale that I'm very reticent to be like, our 550 milligrams of choline supplement, you can back translate back to eggs or whatever, which would be like 5 eggs, a few more eggs a day. So it's not like I go around recommending that either. But our supplements, I really encourage people to interpret it as the effect of the supplement and not back translate to food so much.
Yeah, but most prenatal supplements do not contain choline because they're so big, because it's so much. Is that true?
Yeah,
Ish?
so choline, a lot of our micronutrients are in like microgram or low milligram amounts. So we're giving 550 milligrams. And so
Yeah.
that is like, if you've seen a calcium pill, it's so quite similar in size to that. So you'd have to double the size of the, calcium in prenatals is often pretty low. It's only like 100 milligrams because it's taking up so much weight in that pill size. So you have to have a whole second pill,
Yeah.
basically. It's a
The
similar
same with
issue
the
to,
DHA.
yeah.
Yeah.
It's so funny. I think like I always think about like, there's no like amazing studies for like manganese in pregnancy, but we just throw it in there because it can fit. But because these nutrients are they're metabolically so much more important during pregnancy, but because they're so big, you have to have this like super high level of evidence to ever tell somebody to take a supplement with the D or I level of them. Whereas like, selenium is not really like killing it on the RCT front.
No. It's where it's very, very frustrating. I think there's like one supplement I saw that it's like 8 pills a day to hit all of to get as much closer to what they think the actual needs are. And missing nutrients was choline and omega-3 would be major aspects of that. And that's 8 pills a day, like you said. And when with pregnancy, you're already a little bit nauseous. Like this is, there are a lot of issues. Yeah.
I one time was in a dose response histidine study at Cornell, and I
Nice.
had to take like, I think it was like 18 pills or something crazy at the end. I was so tired of it.
It's the
Uh,
worst.
so I mean, and like pregnant women in the studies are like so motivated. They
Oh
want to
yeah.
do what's they feel very like so helpful or so grateful for our participants because they're just so motivated to the cause of like improving our knowledge of pregnancy nutrition requirements. So I'm pretty sure they would do almost anything we
Right.
ask them to do, but you obviously want to make it as easy as possible, and it's got to be something translatable. Like just your pregnancy research participants might be super motivated, but like when you go to if it's going to inform a population public health recommendation around supplementation, it can't be take 8 phosphatidylcholine supplements a day, and there's too much equipoise about like whether PC is really the same as a choline salt. So you got to use the form that would be most reasonable. So Marie's lab has always used the choline salts, but you can find this is a huge problem with the field when you're doing meta-analyses of like choline supplementation in pregnancy. There's only a few trials as it is, and you have to split between who used PC and who used choline salts.
Yeah.
The data gets noisy and messy and is different in so many other angles. So if there are funders listening, please fund more studies on choline so we can have recommendations that are solid. Although this might be a foregone issue, we can get into it if you want. But
Yeah.
yeah, the companies are already coming out based on the couple studies out of Cornell that have shown positive
Hmm.
cognitive benefits. That I think recruiting women, it's very much like a same issue for DHA. Recruiting people into the study population who want to be in a placebo arm and are okay with it when all their friends are taking the supplemental dose already, you can't really get a low exposed group. So I think we're
Right.
approaching that time, and Ritual has a
Hmm.
choline supplement. Nature Made has one as well, and so choline's on the obstetrics beat right now.
Hmm.
And I think it'd be very hard to recruit in America to have that low choline arm, unfortunately.
Yeah. Yeah. I totally. There's so many similarities in the DHA front at this point. Is there a good biomarker for choline, or does it move around? Is it too metabolized in too many ways, where it's like,
Yeah, it's very metabolized. It's like you can measure the concentration biomarker of just serum choline levels, but that is a composite of not only what you're eating and absorbing, but then what is de novo synthesized, and then what tissues are uptaking. And so what you see across pregnancy is actually choline, even in Marie's controlled feeding study with the low choline arm, and you see it in ours with the true placebo arm where they're eating habitual choline intake for even lower than Marie's lowest arm in the controlled feeding study. Choline is increasing across pregnancy. And that's because the liver is just doing what it can to produce a bunch of it so that the placenta can suck it up and concentrate it in the fetal compartment. But it's the perfect example of, just seeing higher levels of a nutrient in blood don't tell you anything about what's happening at the tissue level, because the choline's increasing across um, uh, pregnancy in the maternal serum, but that's because it's accruing in the fetal compartment, and the maternal liver is actually really stressed.
yeah.
And you see this in rodent data better, where at the end of pregnancy, the hepatic choline levels in the dams or the
Hmm.
moms are much depleted at the end of pregnancy on just like the standard
Yes.
chow that otherwise provides the essential requirement. And so we think that the maternal liver is getting exhausted, and that's part of why supplemental choline is helpful, just to replete mom's the hepatic stores of it and the amount of choline that's there to do all the activities that choline should be doing. And that's sort of
Mhm.
what our data supports, is that supplementing, which I guess we didn't get into the results of
We
it.
haven't yet.
Um,
You can go back.
yeah,
Yeah.
spoiler, no, you go
Yeah.
read the paper, but the choline supplementation sort of dramatically, I will tell you, I was a doubter. Like, I was doing this study, I wanted the
Hmm.
experience, jokes, the pregnancy stuff, but I did not think there was going to be like a huge choline DHA effect.
Yeah.
I'm also just, I'm always preparing myself for the null, and then
Hmm.
happy if it's not.
Hmm.
But the choline supplementation had like a substantial effect on DHA status in a way that was quite shocking.
Hmm.
And so we saw like in the placebo arm that just got the 200 milligrams standard of care, that those individuals started about 6% RBC DHA, which is already high. And I
Hmm.
think it speaks to, we're recruiting women from Ithaca, New York, where there's a lot of
Yeah.
interest in nutrition already. And then they went up to about 7% by the end of pregnancy. We're talking about the red blood cell DHA
Hmm.
level. So they saw like a 1 percentage point increase. And choline supplementation on top of that same dose of DHA took them from around that 6% baseline to around 8% at the study
It's
end. And so
amazing.
like it like doubled the relative effect. Which, I've shown that data to a lot of DHA experts besides yourselves, and there's a lot of four-letter words that come about like,
Hmm.
wow, that's a huge effect.
Hmm.
Uh, and I think it has a lot of makes you think a lot about that nutrient interaction, and how you think about past DHA study done in pregnancy, where it's all done in this kind of choline stressed environment, that to really
Hmm.
get full effects of omega-3s, do you need to add choline. But I think our study is very much internally, we look at it as like this is an important nutrient interaction through this PEMT axis, and that you can get. Yeah, we don't have isotope tracing with DHA to say like, was it just more getting out? Was it less
Right.
being oxidized? But from all the data, we can see that plasma levels of total DHA and PC-DHA are up. With our isotope tracing data, we see increased evidence of that activity of the PEMT pathway being fueled in our placental tissue and our fetal red blood cells. We see that DHA tracked up. So it's like mom seems to have exported more and the fetus has taken it up, which is good. It would have been awful if we had had more in plasma and then
Yeah.
fetus was down.
Yeah.
Everything tends in the direction we want. And so we think it's very much, at least one of the mechanisms that choline is increasing DHA status is by increasing the activity of that PEMT pathway and the hepatic export, which I think implies, maybe Bill, you have thoughts on this, that some percentage of the supplemental DHA that we're giving is getting oxidized in the liver because it's not able to get out. And we'd need an isotope tracer to confirm that. But the question is like where, if we're not giving any extra DHA, but choline's increasing all the status markers, it means in the placebo arm, some of that DHA is getting inefficiently used and probably oxidized instead of exported and incorporated into membranes. And so I suspect that like, fat trapping to some degree in the liver from that low PEMT activity is leading to a metabolic trait that we don't want, which you'd see increased. If you gave it as an isotope, you'd see that enrichment in the exhaled CO2.
Yeah.
But that's another study to be done.
That's another study.
So I have several thoughts. So one thing is it possible? So this was really in the second trimester pregnancy,
No,
right?
second and third trimester.
Second and third. In the first study you did
12 to 16. And so we got them right after you kind of clear the fetal demise window. Or, we were worried about something bad happening and rolled up and then went all the way through to, we have placentas collected at the time of parturition or giving birth. The previous study was just third trimester only.
Right. And they didn't see the effect in the third trimester. So they were like, we think this is actually happening earlier. Is that right? Is that
so there was a choline DHA interaction effect that we saw and that Marie saw in the previous controlled feeding study in the non-pregnant group. And then in the pregnant group where they went in the third trimester when estrogen's already really high and upregulated, we think of that as like probably already we intervened late in the choline sort of stress of it all. And at a time when PEMT is already pretty high in its activity. So we wanted to go earlier, kind of intervene when you're metabolically and hormonally more like the non-pregnant state. We saw that effect. And that seemed to be true, that we're going earlier. So it's not that there's not benefit. Marie saw benefit from choline supplementation in that third trimester for infant cognition. So choline had its independent benefit. We just didn't observe in pregnancy the nutrient-nutrient interaction. But going sooner, we saw it quite clearly.
Right. That's where like the having a higher or more sufficient choline intake improves your ability to uptake DHA where it needs to be. Is that, and it's most dramatic in pregnancy. Do you think that could potentially also be happening in a non-pregnant population and affecting like whatever we think of as bioavailability? It's like that next step of, well, if you're not having any choline, maybe that's part of that interpersonal variation where some people have great response to supplementation and some people don't. Is that maybe even another layer?
Yeah, I would love to look more in the non-pregnant, like women of reproductive age who still have high estrogen. This gets complicated to look at with controlling for cyclicity and oral contraceptives and all these sorts of things. But Steve Zeisel's work, where they followed up from the data that informed the DRI was really just men. Later on, he did data in women, both of reproductive age and also postmenopausal women, in efficiency type study designs, and non-pregnant women who are placed on a deficient diet see a reduction in the amount of PCs that contain DHA in circulation. And so we think that this choline DHA interaction is relevant, but we only know from the non-pregnant state that it's like, make the diet like super, super, super deficient, you compromise the DHA export in sort of a inverse way of what we're looking at in pregnancy. But asking whether like choline insufficiency across a more normal intake range, not in a deficiency diet setting in non-pregnant women, might be a determinant of DHA status, I think is a really valid question. And you could do with everything from, just getting dietary reports and associating DHA or choline intake with DHA status, potentially in like an observational way or in a, controlled trial type setting. But I suspect it would be, it's this complex interaction of your methyl donor requirements with your methyl donor need, your estrogen status and PEMT activity. And so, and you add into that then the other determinants of DHA status, like your baseline DHA. And it would be I think it'd be a pretty big sample size to consider here. I think pregnancy is so metabolically stressful, and there's such a massive upregulation in estrogen driving that PEMT gene expression and activity of the enzyme that it's like such a stress state for most people. That the choline supplementation effect revealed itself relatively well. The choline DHA link in the non-pregnant state, I think in Steve Zeisel's work, revealed itself because you place them under extreme choline deficiency.
Right.
But I think teasing it out in the general population is going to be a much more subtle effect, and so you need a big sample size. And because there's not a choline status biomarker yet, it makes it hard to say like use that as an objective measure. Because with self-reported dietary choline, it's tough because a lot of processed foods use like lecithin, which contain as an emulsifier, and that contains PC, and so that's not captured in a lot of the food nutrient composition databases all that well. And then it's not captured, obviously, when you give a self-report, even if you have a really strong 24-hour recall with a dietitian-led probing. I guess if you do that with an eye towards choline, and we're like reviewing nutrition facts labels to see the emulsifier amounts, or whether there's emulsifier used in the product, you get a little bit better. But we think that there's an underestimation of choline intake in a lot of the American diet because you're just not capturing well that emulsifier use.
Okay.
And it's sort of ironic. It's like if you don't eat a lot of processed foods, you're probably not getting as much choline unless you're eating a lot of egg. And so it's hard to, I think, all self-reported dietary data has its issues for sure. And it's good on some nutrients and not so good on others. And with choline, we don't think it's particularly amazing, but the validation studies aren't well done for that.
Yeah. And I mean, I'm kind of thinking of vegans right now, is because that they'll come and they'll usually have pretty low levels of DHA and then go on to like an algal DHA supplement. But if they're totally vegan, are they getting, I mean, eggs seem to be the main source of choline, but also like seafood and meats, right, are pretty major sources. And so if they're just getting DHA without choline, is that potentially like a stressed state for them? Like
I think that would be the population you'd want to like intervene and study. There's a caveat here. So so we talked about choline being oxidized to betaine and used as a methyl donor. And that is an important metabolic fate. You eat a lot of dietary betaine. And so choline itself is in more plant foods, but choline itself is in more animal foods because the animal cell membrane contains phosphatidylcholine. But you eat a lot of betaine, particularly in plant foods like dark leafy greens and grains. And so it's known from animal work. We cannot put a number on this, but it's known from animal work that betaine can, to some degree, fulfill what we think of as the choline requirement. And so vegetarians and vegans, whether they're all walking around like super choline deficient, they may be partially meeting their requirement through betaine.
Nice.
And so the degree to which that all maths out is that we can't really put a number on it. Vegetarians are still like eating at least a couple 100 milligrams of choline per day. The deficiency symptoms for choline are so nonspecific. Fatty liver infiltration, slight increases in ALT, AST, and a bit more dramatic muscle CPK, like damage marker in the deficiency state. But as a clinician, like for you to say like, oh, you have a choline deficiency, but based on your biomarkers, it's not really there. So it's really tough to say much about dietary choline adequacy and whether specific populations are going to be super high risk or not, because there's not really good national monitoring of this. We're worried about our self-reported dietary intake measures. That's why like a lot of what we've done is we've gone in states where we know metabolically there's a big demand for choline, and we basically like doubled choline intake, give or take whatever the normal variation is. So you'll see the biggest effects there. But whether the NIH is ever going to fund choline feeding studies in these populations, not holding my breath for that one, unfortunately.
No. I'm also thinking about one of Susan Carlson's older studies. They did DHA enriched eggs was in the intervention in their pregnancy population. I can't remember the name. Was it Kudos? I don't remember. But it was like 600 milligrams of DHA. They had a huge effect in those women. And they're also usually low omega-3 intake. And I just think for pregnancy, eggs are a lot more palatable and a lot less scary than fish for a lot of people. But there's also just really strong data on seafood intake and cognitive outcomes for kids. And that's choline and omega-3 and a lot of other great stuff.
Iodine.
Uh-huh. And so the reduction in fish intake due to the potential methylmercury toxicity has just a lot of effects. If that was such an amazing nutrient like source of all these nutrients that are pretty stressed in pregnancy and pretty needed. So there's a lot around. I mean, the DHA train is really pushing that right now, I think, to get that reversed, but it just comes along with like so many other good knock-on effects, I hope.
Yeah. I think that if you're increasing animal foods, you're going to get a solid amount of choline coming in, or it's going to be a decent source of choline.
Mhm.
I always highlight for folks though that in our studies where we're clearly seeing cognitive benefits, it's from a supplemental dose, which is
Yeah.
itself a biochemical form of choline, and it's like whether people are going to be eating 4, 5, 6 eggs a day is pretty questionable. So until
Yeah.
I would love to do like a food-based choline intervention study to see,
Yeah.
does it supply the biomarkers in the same way that we see? Does it influence cognitive outcomes in a similar way? Obviously, you won't be able to attribute it all to choline, but
Right.
there's a big need for. I could go. I have some papers on this I can send you if you guys want to link in the show notes. But just
Yeah.
our dietary data or our dietary intervention studies in pregnancy are so limited.
Yeah.
We have a few of these supplemental studies that exist on really important nutrients like folate and
Mhm.
choline and DHA, but translating that back to food-based recommendations is really tough. There's only a few controlled feeding studies ever done in pregnancy, or even food deliver studies, and
Mhm.
we're worried a lot. We're thinking a lot about like this is such an important time, and but also a vulnerable time. And I think
Mhm.
there's a big push, particularly from bioethicists, saying like pregnant folks need to benefit from research, not be protected from research, because that only leads
Yes.
to in the long run that we don't have data on them. And so if there are again funders listening, like please fund, this is like such an important physiological state that we really are guessing at a lot of our nutrient requirements and back translating data from supplements into food, and it's
Mhm.
a bit of a mess. As you pointed out, like foods are rich in many nutrients and many bioactive components that the effects may be bigger than we predict based on nutrients alone.
Mhm.
So I would we're coming off finishing a controlled feeding study in Tanzania, because it was easier to build a metabolic kitchen there and do a controlled feeding study there than it was to do a controlled feeding study in America. Like, I mean,
Wow.
the infrastructure and the funding here is just so awful. And so I'm always on this social media being like, please, everybody cares about making America healthy and all this stuff. I'm like, well, you got to fund the research to actually
Yeah.
know what is healthy.
Yeah.
Uh,
Okay, that's a
Good point.
perfect transition because I wanted to talk. Sorry, do you want to go ahead?
No, I
Go
think
ahead.
that's a great point. Very timely point.
Yes. And also, so you have this deep research knowledge. You also interact with the public. You go on Instagram and you talk to this level, this like depth of detail about things, which I just love it. And you also have the AJCN In Press podcast as well. So you're translating research, and you're also coming up against misinformation. What's the motivation to do that for you, and how is it going?
I will say a lot of this is just for my own mental sanity, where like, when you're in lab all day running, balancing, I'm got to go to the mouse house and I got to go to cells and I got this Western blot running and like LTMS, I got to go down to this other building to get that. Like, and I was Marie Cuddle's last PhD student,
Oh,
and
wow.
also the last PhD student in Mark's lab. And so I was, particularly at the end, I was alone a lot
Yeah.
of the time. And you can only do so many podcasts and audiobooks that like
Yeah.
I would just, I always liked tweeting and things like that, but
Yeah.
and doing Instagram and whatnot. But a lot of like, it's just when you're sitting there with alone with your thoughts and you're reading the science and you're frustrated about it, I was just tweeting out whatever I thought. Like, uh, so I wouldn't say my translation of science is all that strategic.
Yeah.
Like I'm not, I guess I want to be clear. I'm not trying to talk to them. People always say like, "Oh, you're talking at too high of a level for the public." I'm like, I know. I am just tweeting my frustrations, and I think a lot of it. I think of what my role is like breaking it down for clinicians who are
Mhm.
like informed and want to learn more. That it's an economy of scale thing. Like I could try and be the expert for the public, or I could try and be somebody who's breaking things down for every RD, nurse,
Mhm.
PA, physician out there that knows enough that they want to break through the nuance and make sure they're giving people accurate information. That's sort of that like clinicians as well as journalists are kind of who I have really geared my messaging more towards
Mhm.
to get the right information out there. Because I realized this a while ago. I could tweet into the void about, oh, this terrible study on Splenda or whatever. And even if you get
Yeah.
1,000 people who like your tweet or whatever at that time. I was like, if I can inform a journalist who writes a New York Times article about it
Mhm.
or a Washington Post article about it, and that lives for forever and is a top hit on Google, like that's the economy of scale and impact that I'm more
Mhm.
like one more fit to do and like have the energy to do. And I
Mhm.
think also has a lasting impact. Now that's not to knock on anybody. There are a lot of dietitians out there who give like great
Mhm.
information directly to the public. And I think you need kind of all people walking in all different lanes. But my approach to SciComm has always been like, how do we get a high impact article that kind of cuts
Mhm.
through the meat? Because what you see after you've been in these spaces for 10 years is that the stories repeat over time. The
Mhm.
same limitations of the same study designs come up over and over. And you just say it ad nauseum. And
Yeah.
I think a lot about how do we try and I don't think I'm doing a good job at it necessarily, but like how do we address these things in the long run so that there's at least accurate information out there for folks to get at. I think a lot of it too is like I got into nutrition. I didn't go to medical school. I didn't want to write prescriptions so much. A lot of nutrition's about the flexibility
Mhm.
of things and the nuance of all the study designs and whether it applies to that person that's sitting in front of you at that point is like kind of what you're clinically thinking about. And a lot of study designs have like no relevance to your
Mhm.
patients because it's a totally different
Right.
population, totally different dietary background, et cetera. But a lot of what is out there in the media and what people profit off of is this like relatively dogmatic, highly prescriptive dietary programs, which is sort of like
Mhm.
ruining the fun of nutrition clinically in my standpoint. So over time, I've increasingly just sort of focused on like let's talk about the nuance and poke holes in this because it's not a prescription. Diet is like
Yeah.
so complex and has so many reasons even beyond the science too of why people eat what they eat. But dietary study designs are like just a snapshot of a single context and it's not the same as like a statin versus a placebo that I think leads to a more deterministic prescription for folks. And so getting people to embrace that nuance, let that take away some of the anxiety about like I need to eat perfectly or whatever
Oh,
people are telling them is perfect. Because we're at a point now where everything from carnivore to vegan is the ideal human diet and
yeah.
everything in between. And it's just sort of created a mess of misinformation for folks and stress about eating that should otherwise be like diet is powerful.
Mhm.
But my example for folks always that look at the DASH diet. It's like 10 things that combined they make a pharmacological effect on blood pressure. But any one thing is not like, we it's
Mhm.
important that we have public health guidance on sodium and people think, eat less sodium.
Mhm.
But the sodium effect on that blood pressure lowering is pretty small. The potassium effect's pretty small. The calcium effect's pretty small. The nitrates are pretty small. Like
Mhm.
that's I think reorienting folks to like it is a lot of little things add up to a big thing, and when you sustain that over time, it's meaningful. But we are in the era where every little thing is apparently make or break it.
Yeah, and that leads to so much stress, and I mean, we're pretty focused on fatty acids, we're expanding a little beyond omega-3s now in our research, but, um, like, you study that and then you understand, like, this becomes a part of a diet, you have to get deep. There's still so much we think of omega-3s as one of the cleanest, like, easiest nutrients to study because it has a biomarker, because it has so much data, too much data. Um, Still tons we don't know about it, and
Yeah.
it enters the field of normal people's lives and their diet, and, like, how do you not stress them out, but also be like, this is helpful. Like, this is where pregnancy really, to me, becomes, I want to talk to clinicians too, because pregnant women get bombarded with wanting to do everything you possibly can, but also everything becomes heightened and it's so stressful and you don't know which way to go. And so like if a doctor can help take some of that stress off and see what, like, where do we really, can we really ramp up nutrients in an easy way, in a way that we know a little bit, we know something, but there's still so, like, we don't know everything. But this has been my soapbox for a long time, and looking at being more in the omega-3 pregnancy space versus just a general adult wellness. It's a totally different game in my mind, especially having been pregnant twice, and like being able to have a doctor see, like, we're at a point now where we think we have blood levels that will allow a doctor to prescribe a high or low dose of DHA and that have a meaningful effect on preterm birth rates. And I want that because the fish story is so stressful to women. A lot of women, not all women, it's getting better. The doctor doesn't have time to talk about the ins and outs of fish with the patient because it becomes an argument. So that's where I want like a targeted nutrition, single nutrient, like clean story, but it's also within the context of the entire diet is important. And at the same time, the body's super flexible. We can handle vegan, we can handle carnivore. I mean, you start to push things and things happen, but like that's why studying nutrition is fascinating and the most frustrating thing in the whole world, because it is so, there's just every variable in the world is affecting it.
Yeah, I mean, we talked about my like origins in biological anthropology briefly, but I fall back on that so much of like thinking about the adaptability of the human species across so many environments. And like, yeah, I'm not saying adaptability doesn't always mean optimal, and that's a lot of what we're thinking about is that added little benefit doing, you know, the amount of DHA, the amount of like saturates you're eating, whatever it is, like these are all optimizing, but not doing it wouldn't lead to like a catastrophic effect, which I think is what I try to emphasize. I still see patients, and a lot of the pregnant come to me, and yeah, I'm like, first let's talk about foodborne illness. Like they think I'm going to spend all my time like talking about like which nutrients to eat. And I'm like, no, there's like some big high impact things that are acutely worrisome.
Yeah.
And then we'll get into the like 2 IQ points for your child sort of stuff down the road. But it was really about that triaging and that sort of setting prioritization of the goals, and making sure people are oriented correctly. But yeah, they're not worried about the low amounts of methylmercury in the fish, and kind of talking about the high impact of the omega-3s and the iodine and all these things and what all the cohorts you say. But communicating all that is really, really difficult on a one-on-one level. So I do not envy, I think, a lot of clinicians out there, especially obstetricians and gynecologists who are frontline on this, who want just a soundbite to give, like to tell people who are in passing because they've got 40 other things that they need to talk about in their visit.
Yeah, that's exactly when we've talked to clinicians. And that's the biggest issue is time. And like you just said, they are so much more orientated to reducing risk versus the other stuff, because they've seen the worst that could happen when you don't do that. And of course, that's like the main thing that they're going to spend their time on. So there's like a big disconnect with research and what clinicians have time for and how they communicate with their patients that I think is really important to bridge that gap.
Well, as 2 dietitians here, I think we could be nepotistic and say that there needs to be expanded insurance coverage for pregnant folks to see a dietitian, because we're asking the physicians and PAs and nurses to do something that they're not fully trained in and don't have the time for and don't get reimbursed for. And we have this totally unused clinician population of RDs that I have patients have to pay out of pocket and resubmit to insurance trying to get coverage. And I know oftentimes they don't, just to get like, it's crazy to me that like we're just even a one-time visit to talk about like what's the high priority food safety things, what are the nutrient things. But you know, even later on in pregnancy, I've seen patients that are having GI side effects from the iron and just wanting to think about different dietary strategies. And I'm like, it's so sad that is, we have all these dietitians out there not being effectively utilized. We have all these patients who could be benefiting from that, but there's our insurance model and the coverage model doesn't really allow for it. So there is a Medical Nutrition Therapy Act sitting before Congress.
Oh, really?
Yes, it was introduced in 2020, right before the pandemic.
Perfect.
It still has not seen much action. And so,
Yeah.
there's a lot of hype nowadays about expanding, improving the nutrition of Americans. And it's like,
Yeah.
well, you still, for the most part, have to wait until you have a disease to get your insurance to cover seeing a dietitian. And if we really care about prevention, we need to massively expand access
Oh, yeah.
to dietitians.
Yeah. Amen. We can finish there. We just went way deep and also pulled way out. And that's always a conversation with Kevin Klatt lands in that zone, which is great. So thank you so much, Kevin. You're great. This was a really interesting conversation, and we're excited to see where you go next.
Thanks for having me on. I can't wait to put OmegaMatters on my CV.
That's right. Thanks. All right.
Awesome.